Hello, everyone, and welcome to phyloseminar.org. This is the last talk in a series of 3 talks on next-generation sequence evolution models. Please use the YouTube live comment box to ask questions. Today's speaker is Antoine Koehl. Antoine started out as a biochemist and structural biologist. He received his BS in molecular cell and developmental biology from UCLA, and then went on to do his PhD in structural biology with Brian Kobilka at Stanford University. There he worked to understand the molecular basis of G-protein coupled receptor activation and signaling using structural and biochemical methods. This experience kicked off an interest in learning about expansion of function in protein families that share a common fold. He's currently a postdoc at UC Berkeley, having made the transition to computational biology and machine learning, advised by Yoon Song and David Savage. Welcome, Antoine, and thank you for participating.
Thanks for having me.
And just so it's all here and permanent on YouTube, can you pronounce your last name correctly while I just sort of bungled it?
That's all right. I would say probably Koehl is
Cool.
Probably the easiest way.
Okay, I made it too complicated. All right, take it away.
All right. So let me get started. So the title of my talk today is Deep Models of Protein Evolution. And I'm going to kind of start with a little bit of background and history before I dive into kind of the nuts and bolts of the work we've been doing in Yoon's lab. So kind of the simplest thing you might want to ask is, what is a model of protein evolution? And basically, kind of broadly speaking, a model of protein evolution quantifies how proteins evolve over time. And they're really useful to understanding how evolution has led to a current protein of interest, or they can be used to predict how a protein will evolve kind of in the future, kind of prospectively. For example, as a biochemist, I might be interested in evolution of the amino acids that span a small molecule binding site. I want to understand how they are selected for to give a protein the ability to selectively bind a specific molecule that it can use for signaling, enzymatics, or what have you. And so classically, the way we've parametrized and kind of fit models of protein evolution is by using a statistical tool called the continuous time Markov chain. And the core, kind of what really parameterizes a CTMC, or continuous time Markov chain, is a transition rate matrix that's frequently called Q. And historically, kind of efforts in the field were focused on estimating better transition rate matrices as we got more sophisticated model capabilities, but also as we had access to more and more sequences. So since I'm going to talk about this a fair amount, I want to just give a little bit of background into a transition rate matrix. And so the transition rate matrix really gives you two things. The first is amino acid specific mutation rates. So for example, you could say tyrosine mutates at a rate of 0.66 mutations per unit time. And number two, it gives you a conditional transition probability. And it tells you kind of for a unit of time, when an amino acid mutates, what does it mutate to? And for people who are kind of familiar with proteins, you wouldn't be surprised to know that when tyrosine mutates, roughly half the time it'll either mutate to a phenylalanine or a histidine, because they share a lot of the biochemical features with the parent tyrosine. And an example of a rate matrix would be the WAG rate matrix. This is a fairly well-known kind of classical model. And you see, you can read out the mutation rate based off of the diagonal, which cancels out the rest of the row, and the off-diagonal elements indicate rates of mutation to other amino acids. But ultimately, the whole point of learning an evolution, the way you fit and learn an evolutionary model, is you estimate a joint probability over a set of sequences that you observe in the present time. So here I'm indicating 6 sequences in dark blue, but you have to account for the fact that they are related to each other based off of a tree structure. in that sequence 1 and 2 are more related to one another than they are to any of the other sequences. And a complicating factor is that you don't have access, kind of in most cases, to these ancestral sequences. And that's going to be a big problem. And I will talk about kind of ways that people have dealt with this in the past. So really kind of going far back in history, back to 1978, I would say this is probably one of the first kind of real models of protein evolution that we can really start to understand, or that we can understand kind of the present approaches to modeling protein evolution through the lens of the thought process and the techniques introduced here. So this was from Margaret Dayhoff. And kind of a fun fact, 1978 actually predates the common DNA sequencing methods that exist and that have allowed kind of expansion of protein sequence repertoires. And so a lot of this was based off of direct protein sequencing. And so that kind of gives you a notion of the amount of data that they were working with back then, but also an appreciation for kind of how prescient it was to start developing models to understand how proteins evolve. And so Margaret Dayhoff's approach was the following. And this is kind of a cartoon figure that she brought up, that she shows in the paper. You have pairs of extant sequences that differ slightly. And so in the first step, you impute ancestral states. Here she used the maximum parsimony approach. And then from the ancestral states, along each edge, you can count transitions that happened. So in here, the B mutated to a C. Along this edge, the A mutated to a D. And you go and so on and so forth. You count the transitions here as these points accepted mutations. You normalize them based off of the number. The rate of mutation of any particular amino acid is you count the number of times that it mutates divided by the number of times that it had an opportunity to mutate. And then you aggregate this information across a large number of families. And then here you can get a lower triangular matrix that describes the relative mutabilities of different amino acids. And so this was a really kind of a groundbreaking model, a really seminal work in the field. There were a few issues still. Number one, kind of the imputation of ancestral states might be biased. And there's really no notion of time. You allow for one mutation across each branch, but each branch is roughly going to be the same length. In follow-up work, in 1992, you had a very celebrated model called the JTT model proposed by Jones, Taylor, and Thornton. And their approach was similar to Margaret Dayhoff's, but different in one key aspect. The idea was to completely avoid the imputation of the ancestral states. And so the way they did this is using guide trees, they were able to pair up pairs of sequences on the tree, and we're going to call these cherries. And you look over all pairs of sequences, and you just directly count the transitions between these pairs, between the cherries, the pairs or the sequences in the cherry. And then you do a similar procedure to generate a mutability matrix. The nice thing here is that you don't impute the ancestral states, which kind of adds an extra layer of complexity, and can also introduce bias. The issue, they still had one kind of small remaining issue, and that is that there was really no notion of time. Basically, the cherries were selected for being similar enough such that you could discount the possibility of multiple repeated mutations happening at the same sites. But once again, any two cherries, you could have some that over the entire length of the protein differ by one amino acid, or others that differ by 100 amino acids. And those kind of get roughly equal weights, even though you'd expect more time to happen in the latter case. And then in the early 2000s, you get the advent of maximum likelihood methods. So kind of the celebrated first model of this class was the WAG model from Whelan and Goldman. And here, the problem was just a maximum likelihood estimation to determine the rate matrix that best accounts for data given a series of multiple sequence alignments and trees. And the way that they're able to, and here, rather than doing a hard imputation or like some kind of maximum parsimony, is they use Felsenstein's pruning algorithm to integrate out ancestral nodes. And this once again limits you to independent sites models, where each site in the protein is kind of modeled as evolving independently. And then in the later 2000s, you had kind of the next level up, which is the LG model. And there are various LG models that have been proposed. And the way they kind of get a little bit beyond the independent sites is they can introduce site rate variation. And the intuition there is that, and something that's very obvious when you look at a multiple sequence alignment across multiple proteins, is that different sites in a protein family will evolve at different rates. Some sites experience very stringent selection and don't really vary over time, while other sites are much less constrained and will show a much higher variance. And then there's kind of intermediate levels of selection, maybe for a specific function rather than, say, a fold or an enzyme active site. And so the main the advancement of the LG model was to introduce site rate variation, or in this version that I'm showing the abstract of, you fit a small number of distinct rate matrices, and you find the rate, and you pick a different rate matrix for different sites in families of proteins. And so if we, so that's kind of that was kind of the status of the field into the mid 2010s. And once again, kind of bringing it back to the problem is kind of inferring this joint probability over a set of sequences is actually quite challenging. And it requires, and different groups have used different techniques, JTT, avoid the integrating out the ancestral states. Others find kind of clever ways to go through the ancestral states that we don't have access to. But what recent work in the lab has done that was very exciting, and this is, I think, an alumnus of the phyloseminar series, Sebastian Prio in the lab, published a paper a couple of years ago that recast the problem of inferring this joint likelihood into and introduced a composite likelihood estimation that is going to be consistent with the full joint likelihood under weak conditions. But basically what you do is, instead of having to account for the entire tree structure, you estimate the joint probability as the product of a bunch of conditional probabilities over what are called cherries. The idea here is that you're essentially decomposing the tree into a bunch of trivial trees. And the trivial trees are just pairs of extant sequences, and you can call one of them the root and the other one the leaf. And you're just estimating an evolutionary model along these trivial trees. And so in this work, Sebastian and others in the lab showed that this composite likelihood or pseudo likelihood decomposition was consistent under weak conditions, and they could infer models that exactly matched, the classic models, but with significantly reduced computational burden. So the idea is something like this: you pair up the cherries, and then you can linearize them. And what you end up having is a data set of pairs of sequences that are separated by different times. One cherry might look something like this. Here I'm showing two sequences that are aligned, that you can see a mutation, alanine to serine here, this glutamic acid to alanine, and so on, as well as the time that separates them on the tree. And so the key insight that kind of came out of this paper is that composite likelihood is actually a very general way to recast a lot of these classic evolutionary models. So in this sense, the CTMC-based models that really dominated the field over 40 years comes from, the estimating a rate matrix is basically maximizing a rate matrix that maximizes the probability over pairs of sequences X, Y, T over all cherries. So since I'm going to be talking about cherries quite a bit, the way we like to refer to them is you have a starting sequence X that evolves into a final sequence Y over some time T. And so the rate matrix that maximizes the likelihood over all these cherries is going to be the one that best fits the data. But you can actually, but the key thing here is that you're not really only restricted to CTMCs. Basically, any arbitrary model class that maximizes some likelihood over cherries is going to do a pretty good job of modeling protein evolution. And so this opens you up to kind of much more flexible models. This can include convolutional neural networks, recurrent neural networks, or even transformers, in which the parameter estimation happens by maximizing, once again, the likelihood over a set of cherries. And so really kind of restating this kind of more concretely or with an example is that learning from evolution really just kind of boils down to learning how proteins change over time. And so these are processes in which you have mutations, you have insertions, you have deletions. And the idea is to fit a model that can learn from patterns of evolution across many examples of individual sequences, and ideally over many different protein families to have a single unified model that can share data across proteins and across families of proteins. And so I'm going to now walk through kind of what we did to look into this. And so the way that we use a single data set for kind of evaluating different kinds of deeper models, as well as classical models. And the data set we picked was one that had been previously published because it made it easy to associate proteins, their evolutionary homologs, and their structures in a convenient kind of pre-processed data format. And as a protein biochemist, what I really liked about this was that it came from a sequence clustering of the PDB. And so the PDB, or the Protein Data Bank, at least at the time that this data set was constructed, contained about 200,000 distinct protein structures. However, many of them were kind of the same protein that had multiple kind of structures. Say, as you create a mutant, or you add it, or you have different conditions that you want to get the structure of it in. And so you can do a sequence-based clustering of the PDB in which all structures that share 30% of identity get folded into a single cluster. And so that brings you down to about 15,000 clusters at 30% sequence identity. And from this, we selected 14,500 of these as training families, and the remaining 500 as test or evaluation families to see how well we generalize. And so that's not to say that the test families are going to be completely distinct in terms of their structure from the training ones, but by design, they should share at most 30% sequence identity with with proteins in the training set. And since we're learning an evolutionary model, what we really need to do is get these cherries. These are, once again, the source of everything in this framework. And so from each cluster, you can get the representative sequence. You can we used HH Blitz, or at least the people who, the lab that generated the TR Rosetta data set use HH Blitz against a sequence database, which is just kind of an HMM matching to find evolutionary homologs. This yields a multiple sequence alignment. We use FastTree to calculate a tree. And then the way since we wanted to assess how well the model generalizes at different scales, what we did is for each tree, we found a balanced tree split, which basically boils down to an early edge in the tree that gets used to separate the tree into two subtrees that roughly have similar numbers of leaves. And one of those trees at random subtrees was called the test tree. The other one was called the train tree. And the idea is that you're going to have similar distributions of cherry distributions in terms of the times that separates pairs of sequences. But by virtue of the fact that they come from kind of two halves of a larger tree, it's very unlikely that any sequences in one tree are similar to sequences in the other. That is, there's going to be a maximal amount of similarity between the two subtrees, but they're going to have similar patterns of evolution. And so this test train split within the training families was kind of one level of generalization. But we still have the other splits, which are kind of 500 or 500 clusters that were completely held out. And so I can take, and so what you really need whenever you're doing something like this is to have some baselines. And I can show a few baselines in the format that we evaluate these composite likelihood models. So the first one is a trivial baseline, the random guess model, in which you assign a probability of 1/21. So here we were modeling gap states. So the likelihood of each amino acid at each site is 1 over the alphabet size, the 20 amino acids plus gap. The WAG model gives a likelihood over cherries, where you a product over sites of the matrix exponential of the rate matrix indexed at the source sequence and the target sequence amino acid identities. And the LG model is just kind of, as I said before, the expansion of the WAG model, in which you introduce a rate category for each site. So,
Antoine, do you mind? Can you just remind me? So you mentioned this a little bit before, but can you say more about that rate category? That just comes as part of the LG model, or?
So there are different ways you can do this. You can estimate it from the MSA directly in terms of getting relative site rates based on conservation patterns. But you can also pick a number of site rates. I think the most common is 4. And then that becomes part of the optimization problem, in which you have 4 different site rates that with, I forget exactly the distribution that you select for them. And that
I'm just trying to understand. So what is which happens on your side and which happens sort of pre-baked into the model?
The site rates come with LG.
I see.
I'll put that. I'll say that. So we're not we re-estimated all of these models using the CherryML framework. We fit the LG model with 4 different site rates. WAG is basically LG with a single site rate, where every site rate is 1 uniformly. And then when you evaluate the likelihood of a pair of sequences, the site rate is global to a multiple sequence alignment, and that each column in the multiple sequence alignment will have one of 4 site rates.
Yeah.
And then so across all pairs, say position 20 will always have the same site rates for any pair.
And that's a per codon site rate.
Per codon, yeah, per amino acid, per position, yes.
Yeah. Okay, thanks for the clarification.
Yeah, no worries. And so we can look at, so the first thing, so these baselines are looking at the data likelihood, which is classically the way you would evaluate kind of how good an evolutionary model is, by holding out some amount of data. And so here I'm differentiating between the subtree splits for families that were seen in training, as well as the held out families. And the random guess model, and I'm going to stratify the average per site likelihood as a function of time. So as I mentioned before, we have cherries that are going to have different times separating the pairs of sequences. And so we're just going to stratify our average kind of data likelihood as a function of the time that separates the sequence pairs. The WAG model looks like this. I wouldn't worry too much about the initial rise and then fall. I think this just mostly comes from the way we're binning time and the small number of transitions that occur at very low times. And you can see that, I mean, I don't think there's necessarily much to say here. You achieve really high likelihoods over shorter timescales. And as the time separating pairs of sequences increases, your ability to kind of model evolution suffers a bit. And actually, this is probably a good time to mention what time means in this context. And so this is a mutation normalized notion of time. And so the idea here is that a unit of time of 1 means that in expectation, every site in the protein has mutated once. And similarly, a time of 0.1 means in expectation about 10% of the sites are mutated. And so it's not surprising that as you get kind of far out in this time regime, that it becomes much harder to predict evolution. Because once again, while kind of protein folds are really well conserved, it's been really well known for a long time that sequences can be very divergent, and that there is a large amount of sequence diversity that is compatible with a given protein fold. And so moving on to the next one, by adding site rate variation, you do see a subtle but real kind of increase over the entire time range that you look at. And so here's where I'm going to talk about kind of the main model class that we've played around with as we've embarked on a project to fit kind of deeper models by taking advantage of this CherryML composite likelihood framework. And the idea is that we're parametrizing a conditional transition between sequences x, y over t as the product. This is actually a very standard autoregressive decomposition, in that over all sites in Y, it's the probability of each site is contingent on the sites you've already generated, as well as the source sequence X and the time that separates the 2 sequences. And this might not be super clear, but I can kind of show an example of this in our model, in kind of a graphical overview of our model. And so the reason we went with a transformer is they are really the dominant kind of sequence to sequence modeling deep learning architecture that was originally developed for machine translation. So you're translating between a pair of sentences in, say, German and English. And so you can, in the CherryML composite likelihood framework, you can view evolution as translating between two versions of the same sequence, depending on some amount of time that separates them. And so the way this works is that we have kind of for say this transition, this example transition, X, the context, the starting sequence is shown here, and the target sequence is shown here. And the idea is that we just do the standard autoregressive kind of modeling loss in which we offset the target by 1 by adding a beginning of sentence token. And given this initial beginning of sentence token and the entire contextual representation of the starting sequence, we predict 1 by 1 what the next amino acid in the protein is going to be. And the reason that transformers are really kind of nice is that they split the work into 2 parts. And so you have an encoder, which does representation learning on sequences more generally, but in this case, so while it's training, it'll learn kind of representations across a large number of protein sequences. In this case, it generates a kind of richer representation of the source sequence. And it shares that information with the decoder using what's called a cross-attention mechanism, in which when the decoder is trying to predict site 20 in the Y sequence, it gets access. It gets to see the entire X starting sequence, as well as the 19 amino acids it has already generated. And you can think of it as being by being able to see the entire kind of source sequence, you're able to roughly see where you are in the sequence, as well as what other amino acids have what kinds of mutations you should already expect, given the time. And we also provide time to this model as a sinusoidal encoding method, or sinusoidal encoding, which is a little bit like positional encodings that have been used for differentiating between the first, second, third, fourth position in a sequence. And it's actually very similar to what people use in diffusion models, but there the notion of time is actually quite different.
Can you just continue talking about that for a little while? I mean, I'm familiar with the use of sinusoidal encoding for positional encoding, but here,
it's very similar in spirit. In fact, when we started this project, instead of using a continuous time, we used discrete time bins. And then the idea is that just like for the transformer positional encoding, there is a relationship between position 1 and position 2, which I believe is I think a slight rotation. We set it up in a similar way in that time bin 2 was like position 2, time bin 3 was like position 3. And so you get a notion that the relative difference between 3 and 2 is similar to the relative difference between 2 and 1, but 3 and 1 is larger. And so it's just slight offsets that indicate that there's progressively more and more time that has elapsed.
The model basically backs that out and figures out that, like, if I have a certain sinusoidal, I want to be evolving this effectively evolving.
Exactly. It's a little. This is. I mean, I'll show that these models are very performant, but one of the issues we frequently have is that they are a little opaque, and understanding why they do what they do can be a little bit challenging. But we found, a variety of different encoding techniques, as long as they maintain this property that as you go, that the relative difference between real time, say 0.1 and 0.5, should be reflected in a similar linear relative difference between the encoding of time of 0.5 and 0.1. And it turns out it actually ends up working really pretty nicely.
Thank you.
Yeah. And so this was something that we were really excited to see, is that if you look at the same kind of composite likelihood evaluation over the data, the kind of within family split and the held out family splits, you see that our model, which we call PEINT for protein evolution in time, is by far the best in class. And it actually showed a much larger margin of improvement over the kind of compared to kind of previous to what had been done over between the WAG and the LG model. Another thing that we should note, and actually I feel bad for not going back to this, is that since we're not using a kind of a CTMC that relies on a rate matrix to change kind of equivalent sites, and we're really just recasting this as a sequence to sequence modeling, we can actually do this on unaligned sequences. So we're no longer restricted to a multiple sequence alignment. I mean, so there's a little bit of subtlety here in that, in practice, what we end up doing is we use multiple sequence alignment to build the tree that we use to extract the cherries. But when we extract the cherries, we go back to the original full-length sequences before they were aligned. And so there are no more gaps. There are insertions. There are deletions. And so in this plot, you might ask, you have unaligned sequences now, but you're comparing to classical models on that operate solely on aligned sequences. And what we end up doing here is our transformer outputs a per site likelihood over all sites in the sequence. And we can just subset that over the equivalent positions in the multiple sequence alignments, so that we are making kind of as fair of a comparison as possible, in that we only averaged likelihoods over sites that were present in the alignments. Though we do have, in some cases, a little more information because we do get full sequences. All right, so that's kind of the retrospective evolution, in that we're describing the data likelihood of existing sequences in our data sets. But fundamentally, since our model is a transformer, it is actually a generative model. And so what we can do, and this has been kind of the most recent line of work, is we can provide an initial sequence. This is what we've been calling X, as well as a tree time. So here 0.3. And our model can decode a distribution over sequences that are likely to occur over this amount of evolutionary time. And so we kind of started playing around with this. We figured out different decoding strategies. And ultimately, it turned out that the simplest was what worked best, in that since our model was trained on kind of full-length sequences, we appended to each sequence an end-of-sentence token. That's fairly common in the field of natural language processing. And given the source sequence and the time, we just let the model keep predicting the next sentence until it outputs an end of sentence, and it says, I'm done. I've decoded the full protein. And we found this actually worked very, very nicely. But kind of getting back, maybe not directly answering Eric's question about time. So one thing that we can do is we can fix the initial starting sequence and scan the distribution of sequences that are produced as we vary the time that the model that we give to the model. So the idea is you fix the sequence, you decode a distribution of sequences over time equals 0.1, 0.15, 0.2, 0.3, and you kind of go forward. And you can do a very equivalent procedure with the LG model, in which you just calculate, in which you vary the time that you provide to the matrix exponential, and you can then kind of decode sequences in a similar manner. And what we were really encouraged to see is, so once you decode these sequences, since our model generates insertions and deletions, in order to get a sense for kind of which sites, for the mutational burden that varying time imposes, we can then realign the sequences to the starting sequence and then just calculate some kind of edit distance. And we do the same with the LG model, even though the LG model kind of operates on aligned sequences, so that's actually very trivial. And what we were really excited to see is that as we vary the time that we provide the model along with the initial sequence, as we ramp that up, we increase the mutational burden on the decoded sequences, in a measure that's very commensurate with the classical models. And so that gives us pretty good confidence that the model is actually learning a pretty good notion of time, and that as we increase the time, we can generate sequences with controlled mutational burden that is consistent with the kinds of mutational burdens that we've come to expect from decades of kind of treating these classical models. And so, but what's really important is, you can always mutate a sequence, and it might not be very sensible. But what we can then do is we take the decoded sequences and we use a folding model, something like AlphaFold or OmegaFold, which, given a sequence, will predict a structure for that sequence. And what people have found is that they are also calibrated to output a per site confidence from the model, and that this per site confidence that is usually called PLDDT is actually very well correlated with kind of the ability of a protein to fold. And typically PLDDT will typically go between 0 and 100, but higher PLDDT is going to be better. And so if we look at the sequences decoded, here I'm not showing X as a function of time, but as a function of the sites, percent of the sites that are mutated, we find that our PEINT model generates kind of mutations that are more likely to retain function, fold and function, whereas the LG model does so up to a certain point. But once it starts making more and more mutations, possibly because of this independent sites issue, you see that the quality of the predicted structures drops a lot. And so once we had those kind of sanity checks, so those were on a single branch, in that we have a parent sequence and kind of say a branch length, and we want to see what the distribution over the children of that branch. We wanted to kind of generate we want to see how well we could generate kind of simulations of protein evolution along real trees. And here it's really just kind of scaling up. You just recursively do the single branch step. But you will pick a single sequence for each branch, and that becomes the parent of the next branch. And so we did this over about 1,000 trees, and the topologies and the branch lengths came from real trees. And so these are going to be things that are very similar to trees that you would see in the wild. And we can evaluate kind of the reasonableness of these simulations using things like conservation statistics, as well as the folding models like before. And that once you get to the bottom of the tree, you evaluate whether the leaves are predicted to be things that, a folding model says, this is a sequence that looks like it should fold. And so I'm going to have to start going a little quickly here because we're getting close to the end and I have a few more slides. If you kind of for different families, for the family that yields the tree that we simulate on, we can look at the most conserved sites in those multiple sequence alignments. We can characterize, what amino acids are at those sites, and we can compare. And then once we simulate evolution within that family, we can then compare back the generated sequences to the real multiple sequence alignments and look, kind of the conservation patterns are very similar. It's not the case for LG, even though the LG is given some notion of conservation in its site rates. But I think most excitingly, and the thing that most highlights the difference, is that we can look at the quality of the leaves by virtue, as I mentioned before, of how a folding model like OmegaFold or AlphaFold what confidence it would give that a certain sequence is likely to be, a well-folded happy protein. And so since there's going to be some level of variation in the folding model's ability to, in the way the folding models assign kind of these confidence metrics, what we did is we kind of held out a subtree from the real tree. And here, since this comes from a real tree, we have sequences that come from a real MSA, that come from real proteins. And we can use these as kind of an internal validation for each tree, such that it's the same protein family and it's a similar distribution of times from the roots. And so what you notice here is that for the real sequences, unsurprisingly, the folding models give them all very good PLDDTs above 80, whereas the sequences generated by using the LG model to simulate down a tree are all significantly lower. In fact, none of the, in only one family. So here what I'm doing is across 500 or so of the simulated families, I pick 40 leaves and I predict structures for them using a folding model. This actually takes a fair amount of time, which is why I'm kind of subsetting to only about 40 leaves. And then for each family, each of the 500 or so families, I calculate the average protein per site PLDDT. And so that's kind of a proxy for reasonableness of the sequence. On the other hand, the PEINT model, you see that it's maybe a little bit worse than what's observed in kind of the real sequences. But for the vast majority of families, it is generating sequences that have, on average, a PLDDT above 80. And that's usually picked as a threshold for saying something is going to be confidently folded. And if you compare two different simulation methods, kind of a classical model, LG, versus the PEINT model, in virtually all cases, the PEINT model does significantly outperforms the LG model. And so we think this gives us pretty good confidence that PEINT is actually doing a really, really good job of simulating evolution down in some arbitrary tree. And so that unlocks a really neat feature, in that now we can generate very realistic kind of protein evolution simulation data. And I'm not showing this here, but with similar patterns of insertions and deletions as you would see in kind of the empirical protein families. And so I'm going to wrap up with kind of returning to my roots in the wet lab for a little bit. And so what we were looking for was to see, a folding model is a great proxy for, something is likely to be folded, but proteins can be folded and not functional. And so that is definitely a concern of ours, because it seems that in many cases, a structure can tolerate a large mutational burden before the protein unfolds, but a much smaller mutational burden before it becomes non-functional. And so we focused on an enzyme. This enzyme is carbonic anhydrase. It's a fairly well-known classical enzyme. And what it does is it catalyzes the hydration of carbon dioxide in solution. And it does so through kind of a bound metal in the enzyme active sites. And so we picked a tree, and we picked a kind of extant carbonic anhydrase sequence. We picked a tree that has, kind of actually a pretty deep, this is actually a fairly deep tree. And we ran our simulation procedure, and we wanted to compare it to kind of the other models, the classical model of LG. And you see that starting here, so the root sequence is up here, and by the time you get to the leaves, basically all of these sequences are kind of based on folding models, are believed to be fairly unreasonable. And by eye, they don't look very reasonable either. The PEINT model generates very reasonable sequences, in that basically all of them have folding scores kind of above 85. And we wanted to test, and the reason we picked carbonic anhydrase is we actually had a very good system to, in large scale, test these sequences for functionality. Sorry, totally got a little mixed up. The other thing that I haven't mentioned yet is that the sequences that get generated in our simulation protocol are not just kind of almost like one worry that you might have with a large language model is that it'll just recap existing sequences. It'll basically be like a dictionary lookup where it'll find other extant sequences. And then in that case, you're not really doing anything new. but what we saw very quickly is that we can take all of these leaves and blast them against the NCBI non-redundant protein database, and then look at the percent identity of the closest match. And we find that for the majority of them, and this tail is comes from the fact that when we rerouted the tree, there's a few of these stragglers that are very similar to the starting sequence, which is an extends one, that the majority of the leaves have, the closest identity of any their percent identity to any protein that's been observed is on the order of 45 to 50%. So this is actually going pretty far out into novel sequence space. But when you take these sequences and predict their structure, you find that the model has implicitly learned to maintain the amino acids that make up the active sites, which I'm zooming in on here. And finally, we wanted to evaluate whether they're functional. And so, I'm going to breeze through this fairly quickly, but the idea is a classic one in biochemistry in which you use a functional complementation assay. And the idea here is that you take a bacterial strain, here E. coli, in which you've deleted its endogenous carbonic anhydrase. And you need carbonic anhydrase to generate bicarbonate, which is the output of the reaction, because that feeds into a lot of metabolic processes. And so these E. coli cannot grow in the absence of supplemented carbon dioxide. And the way you can rescue their growth is by providing a functional version of carbonic anhydrase. And so this is a very nice screen for the functionality of our designed proteins or our simulated proteins, because we just have to look at whether giving these proteins in a DNA fragment to these cells allows them to grow in low concentrations of carbon dioxide. And so we did this. We can see growth over time. This was our initial sequence. To provide a reference, we looked at a previously published mutation, which leads to lower but not fully remove. That lowers the activity but doesn't completely kill the protein. And out of the 20 or so sequences that we ordered, we found about 6 of them showed intermediate levels of activity between full activity and this lower activity mutant that has about 5 to 10% of the parent activity. And so ultimately, we've been really excited about this because I think it shows, for one of the goals we had set out was to create a model that could simulate evolution on any arbitrary tree for any arbitrary starting protein in a way that's very realistic. And the fact that when we do this across an enzyme family, here carbonic anhydrase, a large number of the leaves retain the enzymatic activity that they start with, and they're functional despite having very little sequence identity with any proteins that we observed in nature, shows that we're pretty confident we're on the right track here, in that we can generate we can start to play with these evolutionary models because by simulating, you get access not only to the leaves, but the internal nodes, and we can start looking at, how evolutionary paths down the tree differ in different subtrees, while being fairly confident that we're recapitulating the major points of evolution within that family, as given by the fact that we generate functional proteins. And so actually with that, I think it's about time. I'm running a little bit over here. And so I really want to acknowledge a lot of people who have helped make this project possible. This project really started in Yoon Song's lab, following up on work that Sebastian Priaulx had done on the CherryML, which was the main framework here that I described that allows you to decompose evolutionary modeling into a composite likelihood framework. And working with Sebastian has been so much fun. I've learned so much about evolutionary models. Matthew is an undergrad in the lab who is off to grad school. and Bear, who have been kind of the main collaborators on this project. And then a wealth of people in Dave Savage's lab who have been able to help me kind of get up to speed on the carbonic anhydrase assays to really kind of do the final test on whether or not we're kind of our simulation methods are leading to realistic proteins. And so with that, I think that's pretty much it.
Wonderful. I could say that the number of talks that we've had where someone did a cool evolutionary model and then followed it up with a lab assay is maybe 2.
All right. Well, I'm happy to be a part of that group.
Yeah, it's amazing. So I do invite people to post their questions. So this is so cool. Maybe just to start things off, so now you have the growth assay and you have your PLDDT, whatever it is. Yes. So did you see a correlation between the growth rates and the predicted?
Yeah, that's a very good question. And I think this is, so at the time, that's what I was kind of expecting, that things that the folding models we're most confident on were going to be the ones that are, best growers or what have you. But it turns out there was very little. First of all of the PLDTs were really quite high. There wasn't a huge amount of variance in that they were all between 85 and 90. And looking across more kind of maybe the protein design space where people are using different kinds of models to design, say, a new protein sequence, and then using a folding model to prioritize the ones they test, it seems like in general the field finds that you need it to be above a certain PLDDT, that you have much higher success rate if you're above, say, 85. But above that, it's kind of a toss-up as to whether they'll be functional. I think that one of the best ones we had was actually not something that rescued growth, whereas one of the lower PLDDTs did rescue growth. And so it seems like there's a threshold above which you kind of want to be, but it seems like the metrics that, I guess talking to a number of people in the protein design space, it seems like the in silico benchmarks will only get you so far.
Interesting. I love this figure. It's just like, it reminds me of, there's actually this BNAB paper, where they're just talking about how BRCA1 antibodies are all very different except in the binding site. And so they show a sort of similar figure where it's like the proteins are all over the place, and then they're for the active site, I mean, whatever, not an active site, but the equivalent, they're very homologous.
Yeah, and I think a lot of that comes from the fact, and I glossed over this a little bit, is that part of the representation learning we do from the source sequence is using one of these pre-trained ESM models that has been generally broadly trained over a large number of proteins, and that probably has picked up some notion of conservation, at least in enzymes where it's very trivial and that the active sites are always almost perfectly maintained.
Cool. Eli Levy-Karen asks, is it a monomer?
Oh yeah, good question. This is actually a dimer. So I'm showing if we go back here, in orange and purple are the two monomers that make up the dimer. And actually the active site is a composite active site between some residues in one monomer and some residues from the other. And so when we're simulating the same unit of evolution that we would see kind of in nature, which is just along the monomer. And I didn't show this. So in addition to the growth assay, I've purified these proteins and done kind of more classical enzymatic kinetics. The size of the protein based off of an analytical gel filtration is the dimer. So they're still dimers. And even though we're simulating the monomer, I mean, that the constraints of the dimer are imposed on the monomer in evolution as well. And so I don't know if we would, I haven't tried actually just concatenating two copies and seeing how that ends up working. My guess is probably not amazingly well, but this is a dimer.
I'm sorry, that just went over my head. So, okay, it's a dimer, of two identical copies of the same. thing. And you said that you were simulating one of the two and then leaving the other one fixed, or?
No. We're simulating one of the two, and then we're just saying that you get the two copies that self-assemble, basically. But they're both mutated.
Okay.
Yeah.
That seems like the reasonable thing to do.
Yes.
Okay.
But we don't explicitly, at simulation time, inform anything that there is,
Oh, I see. So you could simulate the dimer.
We could potentially simulate the dimer.
That'd be complicated.
That would be complicated. Another thing, just to see how well the model uses context, is we can potentially give it a context of make a certain number of mutations in one, and then see, given that it knows these mutations have happened, in the second copy, what'll it do? But we haven't tried that yet. So.
Okay, Eli is also asking, maybe the IPTM is more informative than PLDDT, and I don't know what IPTM is, so maybe you can inform us.
I forget exactly, but I believe IPTM is looking at a TM score, which is kind of a structural consistency score across a dimer. Is that correct?
Well,
it's like an interface predicted TM score. So we didn't actually look at that one. I guess the way we had set up the experiment that we wanted to do was actually to not do any filtering. So we actually didn't do any filtering of the sequences. So there were 40 leaves. We picked 20 of them at random and ordered them because we wanted to just say, kind of in your average case, kind of when you get to the bottom of the tree, what fraction of things are folded and functional. It is totally possible that the ones that don't rescue growth are still folded proteins, but they might have lost their ability to carry out this reaction. But we haven't seen that yet. We haven't actually looked at that. So.
Cool.
But yeah, that's a good point. For dimers, IPTM might be a good metric to look at. So.
Cool. Okay, I get another couple of questions, hopefully. So this work is not explicitly structural. Like you have you're just looking at sequence evolution. And if you look at the evolution of things like ESM, they started by using just sequences, and then it's like sequences and structure. Is there any world in which you would... So I know that not all proteins have structures, and yes, we can predict structures, and that gets a little bit crazy. But
yeah, that's a very good question.
How to use structures in this?
Yeah, it's a good question. And I mean, I don't think I'll be able to give a fantastic answer, but kind of so. There are, I guess, different levels to the answer. Number one, because, natural language processing and just general work people have done on transformers, it's fairly trivial to add, say, a structure encoder module on top of that, and that's another thing you can add. And we've been interested in looking, kind of adding structure as a covariate. There's no reason. I mean, it's a little bit of a you can't stop me from doing that, so I might as well try. What I've often wondered is just kind of the sequence to structure models are kind of, you start with the sequence, you predict the structure. Should that information still just already be in the sequences themselves? And so, I mean, I think it's definitely something where we've been interested in, and I think kind of as more students kind of get interested and take on this stuff, I think that would be a very interesting avenue to look at. I would say probably the most simple, like the kind of the intermediate, is using something like a structure alphabet, like FoldSeq uses. And that's very similar to what the newest ESM models use, in which use kind of quantized structural representations. But there's no reason kind of not to do that. And we'd be kind of, I mean, it seems like in the past, prior work has shown that if you provide structure as a covariate, it makes the models the models do a much better job, I think, of decoding sequences. It gives them a lot of good information. And so I think that's probably the next iteration of what we end up doing.
Cool. And I just have to follow up. I still just like this continuous time sigmoid. So you described how you did it in the bend case where you have like several different bends of timing, but after you moved on, I was just like, no, wait, I don't understand. Now you're treating like time as being continuous, but somehow you're still getting a sigmoid. That's just the like.
Yeah, it's been a while since I implemented it. I can definitely give the references for kind of exactly mathematically what's going on. So this comes from the diffusion model literature. So in diffusion models, it's kind of decorrupting, noising forward process that goes from time equals 0 to time equals 1 for unnoised to fully noised. And time gets provided as a covariate to that model, to that class of model as well. And I think the idea was very similar to the kind of discrete time or quantized time case, in which it's a little bit of a convoluted procedure. You generate frequencies, geometrically spaced frequencies, and then I think it's something like the even indices of the representation. You take the sine, the odd, the cosine.
Okay, so it just goes into that.
It just goes into that.
I see.
And the main thing we wanted to do is just kind of, when we generate the initial frequencies, we wanted to find a range that would give us kind of a fairly linear response over the time frame that we expect to see within these evolutionary models. And so a lot of the some of the cherries go up to times of 3 or 4, but you're very unlikely to see something that goes like 100.
Yeah.
And so it was really just kind of scaling to have a linear range across the times that we expect to see, or that we empirically see.
Yeah. I mean, like I said, I mean, it seems like a reasonable thing to do, I guess, given all the other stuff. But boy,
yeah, some of the time as a covariate has been kind of an interesting one. I mean, I think I did skip this. I think I skipped this slide, but let me actually see if I can reshare this really quickly, because I think it was kind of a neat way to. Share one last thing. So, can you see my slides?
I just put it back on. Yep.
Okay. Yeah. So the idea is, so with any classical model, you can just find the time that maximizes the likelihood of, this pair of sequences. And it's kind of, what time is consistent with the mutational pattern you observe. And we can do this with the PEINT model in that we just kind of calculate the likelihood of a pair of sequences as we scan over times. And then you can see that it maximizes the likelihood. And here's a case where it's a little bit longer than the time that WAG has estimated. But once again, we don't necessarily expect them to be identical because we're working with unaligned sequences. There could be an insertion here that is relatively large that WAG might not know how to deal with very well. But in most cases, it's very close. And the shape of this function is actually fairly convex in that there's a pretty clear global minimum. And if we do this across, so what we end up seeing is that for different families, if you compare the WAG kind of time maximum likelihood estimate versus our models, you see that they're very consistent. Though in some families, it seems like there is a systematic offset. And we're not sure yet. I mean, I think this is one of the classic things with evolutionary modeling. You see something different. Why is it different? Is there something fundamental about the biology? Is this a family that is maybe more strongly conserved than you would think? And so I would say in general, the kind of the sum total of the fact that when you vary forward time, you get similar mutations as the classical models, and the fact that that using this kind of this maximum likelihood estimate of the time separating a pair of sequences that is very in line with what the classical models estimate, that even though we're using a pretty simple time encoding that's really just kind of off the shelf, it seems like the model uses it actually as a pretty useful covariate. And that was something we were kind of happy to see, just because we weren't sure if that was actually going to work. But it seems like across the various cases we've looked into, it works really quite nicely. And so it could both be used to help do tree reconstructions, but it's also a good way to kind of generate new sequences with kind of controlled novelty, I guess, as a dial. And that's always kind of fun to have.
Yeah. Super cool. Well, we better wrap. I mean, I could probably continue for a while. I just did want you to say while we're online, the SDSM model, I don't actually know that. Can you say what that stands for?
Which one?
Or didn't you say that there was another model, it's called SDSM model or something like that, that was, it was like maybe an alternative architecture? No? I think I mixed something up. Okay.
We don't know SDSM.
Okay. It was something about, you said that the blank models also do this time, like,
oh, diffusion.
Yeah, but then it was, and then you said people, or no, maybe it was when people are looking at structural. Oh no, you said it was with reference to using a structural alphabet.
Oh yes, fold-seek.
Yeah, but then you said all other models are also using this.
Oh yeah. So the latest ESM model,
Oh, okay. You just said the latest ESM models. Yeah. It's not, and they don't use fold-seek on the nose, but they have their own sort of fold-seek like alphabet.
It's fold-seek like alphabet. That's just higher dimensionality.
Okay. I just the latest ESM models. Yep. Right. Okay.
But yeah, no. So I think, and that's a simple way to just add another sequence to sequence type thing without kind of fiddling with different structure encoders.
Right. Yeah.
So
marvelous. Okay. Thank you so much, Antoine.
Thank you for having me. This was fun.
Yeah, really fun. So our next series is going to be about phylogenetics and public health, and hopefully it's going to start in June. There's been a little bit of chaos because of all the chaos that's happening in public health right now. So thanks again, and hope maybe we'll have you on again in a decade or so. We'll see.
I'm always happy to come back. All right. Bye.